Okay, so good morning, everyone. Thank you for your presence here today. My name is João Henriques. I'll be presenting this with Mafalda. During the next couple of minutes, we'll be talking about performance enhancement strategies for low solubility API and how we can extract maximum value from a potential treatment with reduced development efforts and time. Now, in summary, we'll be showing you how we get from a crystalline, low solubility, poor bioavailability API first into an enabled solid dispersion formulation in 6 weeks' time with 1 month stability and less than 5 grams consumption of API directly to clinical supply without at-scale development and no consumption of non-GMP API, and finally to a successful NDA using 2 less years of development than its standard strategies. Now we'll start with a general introduction to two problem statements and then an overview of amorphous solid dispersions. This will be followed by a technology selection phase and then going into the main content of the session, which are the tools and methodologies that enable accelerated development with reduced time to clinic and time to market. Now, the first challenge we want to highlight is related with the increased pressure to reduce time to market of new chemical entities. This reduction has many positive impacts on both the success of a program and patients' health and patients' lives. This has been recognized by regulatory agencies as exemplified by accelerated approval processes by the FDA in specific cases. The average time from first in human into approval can be reduced by more than 3 years when compared to standard approaches. Not only the number of accelerated approvals has been increasing over time, but also the median approval times when expedited methods are used has also been decreasing. All of this puts increased pressure in CMC, which can become a bottleneck in the overall process if not handled properly. And this pressure becomes even more relevant when we couple this with the need for enabled formulations in challenging drugs. So the second challenge we discuss, coupled with the first one, is the solubility challenge. So vast majority of compounds in the pharmaceutical pipeline present poor aqueous solubility, about 70% of which have dose solubility limiting, which are BCS class 2. This means that enabling technologies are required for an effective delivery. Now, the current formulators toolbox for addressing this has evolved significantly, where lipids and solid dispersions have emerged as the most relevant solutions. Now, in the end, the best technology is obviously the simplest one that provides the results you need. But for most BCS class 2 drugs, simpler technologies such as size reduction, jet milling, or nanomilling are usually not enough, as this mostly improves the dissolution rate. So the choice then falls into more complex formulations such as amorphous dispersions, lipids. The final platform has to take into account the molecular properties of the drug, and so on. There are multiple tools to aid in the selection of the optimal enabling platform. So the use of classification systems, such as the DCS shown here on the left-hand side, modification of the BCS, can help define the level of enablement requirements. So differentiating between size reduction and solubility enhancement. Guidance maps, as the one we use at Ovion shown here in the middle, correlate required efficacy shown by the required exposure shown by the dose number correlated with log P. This can be a useful tool to differentiate between lipids and solid dispersions. Finally, given the number of clinical and commercial programs using enabling platforms, statistical models can now be started to build and using different molecular descriptors to compare a new chemical entity with drugs that have been successfully formulated into either lipids or solid dispersions successfully in either clinic or commercial. In this presentation, we'll focus on amorphous solid dispersions, specifically by spray drying. Amorphous solid dispersion is a well-established platform to address solubility challenges. It is used extensively in BCS classes 3 and 4, shown on the chart top right. It has been approved in a large number of programs, both EU and US. Amorphous solid dispersions are essentially an homogeneous, molecularly dispersed system of an active ingredient and a stabilizing polymer, one or more. The lack of 3D order structure leads to higher thermodynamic activity and an increase in the aqueous solubility. It can be several-fold regarding its crystalline counterpart. The ASDs will typically exhibit a spring effect in this solution, leading to a supersaturated solution. The use of a stabilizing polymer not only maintains the metastable amorphous form of the drug in its solid state, but may also prolong the exposure window by inhibiting nucleation of the supersaturated solution. There are multiple commercially demonstrated processes to generate amorphous solid dispersions, here highlighted in the bottom left, so spray drying, melt extrusion, and co-precipitation. And the number of approved therapies of drugs delivered in amorphous solid dispersions is increasing every year, with multiple successful programs being approved very recently.
Bearing all the challenges and opportunities that were raised by João, we'll now dive into the current strategy in place at Ovion, based on the premise big data, low consumptions. The approach defined consists in a combination of 3 relevant steps. The first, physical stability, and the goal here is to ensure adequate selection of components for the ASD, considering miscibility, avoiding phase separation, and also recrystallization. The second is in vivo performance. Along with a stable formulation, it is imperative to modulate and guarantee adequate in vivo exposure and understand what are the mechanisms underlying this enhanced performance. And in the end, guarantee lean manufacturability through right first-time formulation screening, process selection, and ensuring, obviously, scalability. So let us present us Ovion proprietary screening platform, ASD IPRES, a tool that is able to take a low solubility API and deliver a ranking of up to 24 different formulations. This is based, again, on a 3-step process that starts with in silico screening, based on thermodynamic, drying kinetics, and physical stability to identify the most promising combinations of polymer, API, and surfactant. The second step consists in the production of up to 24 spray drying powders on a modified lab unit. And finally, the last step consists in the analytical characterization right outside production and after a 1-month stability study at 40 degrees Celsius and 75% relative humidity. This analytical characterization, since we are talking only about a screening platform, is limited to determination of the TG by DSC and identifying any thermal events such as onsets of crystallization, and also is based on a 2-step physiologically relevant dissolution method to evaluate in vitro AUC and Cmax determined, both at T0 and after 1-month stability data. And all it takes is 5 grams API in the beginning, and we'll be delivering a ranking of these 24 formulations based on real data and considering in vitro performance and physical stability. So going a little deeper in the technical part and starting by the in silico approach, there's a combination, a combined evaluation of thermodynamics, components, diffusion, evaporation rates, based on the Tenaris proprietary platform and other available commercial models. This way, parameters like excipients ID, drug loads, and component miscibility are identified, and the needs of lab experimental procedures is eliminated, lowering material consumption and lowering as well the risk of going to production with unsuitable formulations. Manufacturing is performed on a lab scale unit under standard conditions, followed by a secondary drying step to ensure the removal of organic solvents. And then the analytical characterization takes place. It will provide the insights of thermal events, as mentioned before, like TG value, the absence of crystallinity, or phase separation, and the biorelevant dissolution to evaluate the degree of enhanced exposure provided by the formulations produced. This data is generated, as mentioned, right outside production at T0 and after 1 month stability data. The total turnaround time for this screening approach is about 6 weeks. So by now, we are able to make an educated decision based on real data for the most suitable formulations to carry on for adequate process development. Our common approach is to select 2 lead formulations, the most promising, and invest then in process development.
So in terms of process development, fundamental understanding of the spray drying process is essential for developing a lean, robust process with minimal testing and API consumption. So our approach to spray drying development relies on 3 main dimensions. The first one is a thermodynamic model of the spray drying operation based on a heat mass balance. This enables the manufacturing space of the spray drying to be defined solely based on the spray drying characteristics and the feed solution without any trials at scale. Secondly, we have an optimization model that relates feed solution properties, optimization system, and process conditions with droplet size. The droplet to particle ratio is a scale-independent correlation, and therefore this can be used for a seamless scale-up between small, large, and medium-scale units. Now, the combination of these 2 previous models gives us combined with a particle formation mechanism, gives us a drying kinetics model, and the drying kinetics is essential to predict additional properties such as morphology, density of the particles, and even in some cases phase separation of amorphous solid dispersions. Finally, all of this can be supported by computational fluid dynamics, not for routine operation, but for optimizing units and troubleshooting very challenging processes. Now, the fundamental understanding in the form of a spray drying model is complemented with additional tools. First, a database, in our case, with over 1,500 experiments, small, large scale, with different solvents, polymers, and APIs. This provides an immediate identification of risks and feasibility of new similar projects that come along. Secondly, scale down tools to generate representative material in the lab with low API consumption, finding the edge of failure of processes, and establishing scale-independent correlations. Now, all of this enables the definition of a robust process for any scale at the lab, with some additional tests that would include, for instance, stability hold time studies, scale-independent correlations using scale-down SD models, which also enable an understanding of the impact of the different attributes of the spray drying material in, for instance, the processability, dissolution, tabletability, and so on. This can be especially critical during early development because the material generated in a lab scale dryer is typically very small, very low density, and dissolves very well. Last thing we want to happen is after we scale up, find out that when we produce the material in a larger dryer, not only it does not get into a tablet well because it has low compactability, but also it does not dissolve as well as expected. So anticipation of risks is essential using these types of tools. All this combined bring us to an integrated platform where with reduced timelines, minimal API consumption, no trials at production, GMP scale. An integrated spray drying and drug product process can be industrialized with reduced risks for scaling, for clinical supply, going up to possible validation and NDA submission. So we end up with a scale-independent design space, representative material in early stages, avoiding the pitfalls mentioned earlier, and a seamless scale-up across all required clinical stages to supply any changing requirements from the clinic.
Analytical characterization of the manufactured powders is performed on a robust and expedite way. For that, several standard methods are in place and available that are product independent. I would like to highlight the GC method that not only will monitor the residual solvents content, but also will mark some of the preservatives of the residual solvents used that are commonly identified upon GMP release as unknown peaks, and this eliminates or prevents deviation and complex investigations when we are under GMP. The only method that indeed requires development, if not yet available, is assay and related substances. And here we will be relying on a quality by design principle. LC method development follows a standardized approach that leverages from prior knowledge, and the definition of the method's operational design region is set along with the method's capacity. An important parameter to guide in the future the interpretation of performance data is the determination of the amorphous solubility of the API, and we do this by identifying the concentration at which liquid-liquid phase separation is attained. And here we will be able to identify at which concentration colloid formation is expected to occur, and then benchmark the dissolution results. Regarding the performance characterization, a detailed understanding of the underlying mechanisms that promote bioavailability enhancement in spray drying powders and can be achieved. As described by several working groups now, upon powder dissolution, several species can be formed in the dissolution media, as seen in the top of the slide. We can see free drug-rich colloids, precipitated drug, and polymer alone. Knowing LLPS will indicate us if drug-rich colloids can be formed, and the importance of their formation relies on their ability to act as a reservoir structure that, upon free drug absorption in the GI tract, it actively will pump out new free drug to the media that will be able to be absorbed as well. And this is an important mechanism for enhanced performance and enhanced bioavailability in vivo. However, our in vitro approach to performance characterization relies on a two-step physiologically relevant method that is able to discriminate total API dissolved and free drug. As seen in the graphs below, for the two lead formulations here in study, they differ in their in vitro AUC, so the total drug available for absorption. And we also did the determination of this colloid's size and stability, as seen by the gray dotted line. Typically, these structures have sizes between 100 and 500 nanometers. And the fact that their size here isn't varying much allows us to infer on their stability and on their ability to in vivo be acting as these reservoir structures that will be pumping API and promoting an extended period of absorption. In the end, it is possible to understand and correlate process parameters and performance and infer on what impacts most this enhancement, either being the drug load, the particle size, or the particle morphology. At Ovion, a proprietary method for predicting the onset of crystallization of amorphous materials has also been developed. This mathematical model incorporates information on the kinetics of relaxation of amorphous materials at a defined temperature, and not, typically the temperature for which is expected to be the storage conditions of the material, and thermodynamic parameters that cannot be disclosed. The model is based on real data gathered from a couple of simple DSC runs. To demonstrate the potential of this method to predict onsets of crystallization, a stability study was conducted at 30 degrees Celsius using films of amorphous drugs and solid dispersions prepared by quench melting in aluminum foil using a hot plate. 6 drugs, as mentioned in the table below, and 3 physical mixtures with different glass transitions and melting temperatures were selected, and for one of the drugs, nifedipine, solid dispersions were also prepared with different polymers and drug loads. These films were kept inside an oven at 30 degrees Celsius for 3 months, and at defined time points, a sample was redrawn to evaluate crystallinity content by XRPD, so real crystallinity data. As seen in the table below, the level of agreement between the results rendered by the stability study and by Ovion's proprietary model is very high. And this model performs better when compared with the KWW model described in the literature, and doesn't require any assumption of a specific percentage of relaxation for the determination of the onset of crystallization.
So thank you, Mafalda. And after we know that our STD is stable, so after we get a stable, enabled, scalable formulation, we go to defining a final dosage form. Now, tablets are usually a preferred option, added to the known advantages, the gelling nature of some stabilizing polymers used in spray-dry dispersions may cause additional gelling issues, which will lead to poor or variable exposure in vivo. But the tablet formulation can be significantly expedited and not be an additional development bottleneck using database approaches. An example provided in this slide. So starting with the input material characterization and coupled with the formulation database, platform-based approach can be used as a starting point. Then using excipient models based on the properties of the pure excipients, these prototypes can be further refined. Finally, a reduced number of viable prototypes produced using an experimental design. The main goal here is to reduce development time, material consumption while maximizing STD loads, improving the performance of the spray-dry dispersion, and also ensuring that we have a scalable formulation very early on. Now, in this slide, we can see the biorelevant dissolution performance as described previously by Mafalda of a bare STD material on top. Now, this is again a formulation with a colloid-forming drug that reaches a very high supersaturation ratio. When adequately formulated into a tablet, and you can see the biorelevant dissolutions results below, the impact on dissolution is inexistent or perfectly residual. The formation of a stable colloidal species demonstrates that the dissolution mechanisms are not affected, and that a proper formulation can at the same time be scalable and perform adequately in terms of potential for exposure. Now, similarly to what we showed for spray drying, the development of tablet formulation and process in the lab may be optimized to extract as much information as possible as early as possible and with as little material as possible. The use of tools such as compaction simulation, empirical or mechanistic models for roller compaction means that the final drug product process can be mostly established using only lab tools. This reduces both development time and material requirements. Scale-up can be made mostly directly up to commercial scale, and most process development, which in the end is always required to support the filing, can be managed and can be done at later clinical stages, and can be done at the final scale where the commercial supply is expected to take place to avoid unnecessary development and process optimization. Now, in summary, and for getting into the clinic for a first-in-human study with an enabled, stable, and scalable formulation. An optimized SD spray drying screening platform for amorphous solid dispersions enables an optimal formulation from 24 prototypes with less than 5 grams API in 6 weeks' time, already accounting for 4 weeks accelerated stability. Process development can be made solely in the lab. The use of computational models, statistical methods, scaled-down units provide a scale-independent process development, which then enables a seamless scale-up to both GMP manufacturing, clinical stages, which can be finalized in less than 4 weeks. For the next stages, the scale-independent process development allows a very smooth transition between scales to suit the changing clinical needs as the program progresses without any scale-up trials or additional development, all of which can be delayed to the later clinical stages when the process reaches its final scales and data is required to support the application to regulatory authorities. So, finally, does it work? Here we have a very small case study which followed all the steps highlighted here. Low solubility API was formulated into an STD formulation with enhanced performance and into a scalable tablet formulation. Formulation and process decisions were driven by a relevant method previously described, which demonstrated an excellent correlation with the PK data, which finally supported going into the clinic in very reduced timelines. At this stage, currently executed 9 integrated programs with this approach for first-in-human, with 100% right first-time approach, no reformulations, ready to support 2 phase 3 studies with the final formulation we derived from this approach. So, concluding, during this presentation, we showed a set of tools and best practices to address the accelerated timelines of pharmaceutical programs, coupled with challenging low solubility compounds. It can be divided in data and science-based methods and computational tools, standardized and state-of-the-art methodologies with scale-independent development, phase-appropriate and selective development, delaying full-blown testing to later clinical stages and the final intended scale for the process. In the end, the final goal is to enable right first-time scalable formulations, reduce time and low API consumption, avoid reformulations along development, and the need for bridging RBA studies, and a seamless scale-up to address growing or changing clinical needs. That is it from our side. Thank you very much for your attention, and I think we can take a few questions.